Lecture 17, and today we're going to look at molecular phylogenies and sequence alignment. This lecture might spill a bit into next class. There's quite a bit to do, although we're well set up now with our understanding of Markov models and models of sequence evolution. I want to say a few more things about evolutionary events beyond single nucleotide polymorphisms that we looked at last class. And I'm going to just talk a little bit about things that I'm interested in terms of how these models also play a role in understanding cancer, specifically tumor genesis and tumor progression. And then that leads into the concept of molecular phylogenies for species or genes. And I'm going to show two different ways to estimate phylogenies. One is via a technique called parsimony, and the second is a fancier technique that builds on our Markov models called maximum likelihood. And then I'm going to finish showing how we can use entities like our PAM matrices to compute sequence alignments, in addition with an algorithm called dynamic programming. So last lecture we saw over and over again the concept of a mutation, for example, from T to C over some period of evolution. Okay, and often, especially in human and clinical settings, we call these mutations, these point mutations, single nucleotide polymorphisms. So really, yeah, there's two different things there. There's mutation, and sometimes they're called point mutations because it's a specific genomic locus, one nucleotide, a point, I guess. And single nucleotide polymorphisms is the other way that we refer to them. There's other events, though, that are very important in both normal physiology and in disease, and in terms of evolutionary, how organisms evolve. One of them is the insertion, and the other is the deletion. So these are generally small events, but not as small as a point mutation. So, for example, we have a chromosome and then some sequence ACGG, etc. And what happens is that this sequence TATA is inserted right after ACGG. Now, I won't go into the molecular mechanisms as to what facilitates those insertions, those short insertions, but it has to do with, often, at least in normal physiology, it has to do with the DNA replication process and mistakes being made. Insertions often, and deletions often, occur around in highly repetitive regions, for example. But yeah, so you get this insertion TATA, but otherwise the chromosome stays the same. The converse of the insertion is the deletion. So, for example, I have ACGG. These two bases here might be lost, and so this A and this G become adjacent in the new chromosome. So these kinds of mutations, insertions and deletions, aren't very long. They tend to be around 1 to 30 base pairs, maybe slightly longer. Maybe there's some that are 100 base pairs or so, and maybe very few that are 1,000 base pairs. They've been relatively well studied, and they often occur, these insertions and deletions occur in regions of the amino acid or nucleic acid sequence that eventually code for turns in the protein structure, rather than, say, in the middle of an alpha helix or a beta sheet. So they occur in certain regions that are more, let's say, forgiving or disordered. And secondly, they often occur in domains, or they're like, you see the complete insertion of a new domain or a deletion of a domain. And we haven't talked much about, or really at all, about domains in the course, but we should try. We're going to try to come back to that in a little bit. The idea is that there are, let's say, pieces of proteins or fragments of amino acid chains that code for different domains, and usually these domains have specific functions, and those domains can be, between 90 and 150 base pairs long, right? And sometimes, shorter. They might be, for example, a single domain might be a type of active site, or it might be a certain kind of structural fold, or it could be it could harbor, say, phosphorylation or ubiquination sites, etc. Okay. If we zoom back out to the genome level and we don't focus too much on a gene, we might see something like this, where your typical diploid, like a human, where you have two copies of each chromosome, and what you can see sometimes are things like aneuploidy, where you gain an entire chromosome, or even more than one chromosome. But for example, in humans, we have triple X. Some individuals have 3 copies of the X chromosome, for example. And you can also have euploidy, where only part of the chromosome is amplified. That's fine too. Or you can have full chromosomes copied. So in this case here, if this was one chromosome, the 2 copies, and now you have an aneuploidy event, again, aneuploidy. Now that person would have 47 chromosomes. That's usually not a good idea for humans. And aneuploidy loss is also possible, so we lose, for example, 1 copy of our chromosome, and then we would have 45 chromosomes instead of our normal 46. Like I said, for aneuploidy gain, you can also have euploidy loss, where a fragment of this chromosome is lost. Okay, and here, when a fragment is lost, for example, if there are SNPs, so these events are compounded in the sense that they can affect each other. So if I have let's say a polymorphism, up here, okay, there's a polymorphism there. I don't know if you can see it, it's yellow, and a polymorphism here that's orange. So this allele is different than this allele, and this allele is different than that allele. If I lose that region through this this loss, right, of those 3 genes, then basically this part of the chromosome will now be connected to this part. You'll still be diploid, but you'll have lost heterozygosity. So the difference between this allele and this allele at both this position and this position is now lost. Okay, so that's called a loss of heterozygosity. You can also have pure loss, where, for example, both copies of a region of a chromosome are lost, or in fact the whole chromosome is lost. That would be a classic aneuploidy. And thirdly, we can have duplication. So, for example, here I have from the chromosomes, from your classic chromosomes up here, we have gene 3, 4, 5 that become duplicated. So this region just gets copied like that. In fact, it doesn't need to be adjacent. So some duplications can occur where some duplications are of the form where this region here is just hopped to an entirely different region downstream or upstream. And as we know, actually, we've seen duplication already in the context of breast cancer, where we talked about ERBB2, or that's the name for HER2. And we know that, for example, this HER2 gene is flanked by GRAB7 and other members of the HER2 amplicon. And that region on, I think, chromosome 17 can be amplified many, many times in breast cancer. And because ERBB2, HER2, is a growth factor, having 30 or 40 copies of that gene, there's a lot of duplications, not just one, but maybe 30, 40 of them. You get massive expression of this growth factor, and that's not a good idea, of course. We all have such SNPs and CNVs, even in healthy individuals. Now, I don't think we're missing an entire chromosome, but we definitely have polymorphisms that separate us, small little point mutations. Now, likely the vast majority of those SNPs are non-coding, so they don't manifest themselves in any way. The copy number variants can also occur in just junk, let's say so-called junk DNA regions, but they might also affect coding regions, and they probably they likely do have a role to play in what makes each of us unique and different. In cancer, though, we have literally crazy things going on. There's an elevated number of point mutations, an elevated number of insertions and deletions, of gene duplication, of gene loss of heterozygosity, and all sorts of chromosomes and other things that are just basically representative of chromosomal instability. So we have copy number change, which we just talked about, CNVs, but we also have interchromosomal rearrangement. So this is a so-called Circos plot, and the chromosomes are arrayed in a circle, not surprisingly. And basically the way you read this is, if you look at this line here, it says that a region of chromosome 13 is now been, in a sense, fused with chromosome 1. So now basically the whole, now you have this kind of hybrid, weird chromosome 113 type thing. So you're bringing genes that are normally very genomically distant from each other into close proximity. In fact, sometimes in such close proximity that these genes that are foreign, let's say those genes from chromosome 13 that are now beside genes in chromosome 1, they take on the promoters that were there for chromosome 1, and the wrong promoters, the promoters could express those genes at the wrong time, especially if those genes from chromosome 13 are, let's say, growth factors. Okay, so, and you can see here that there's not just one or two of these events. There's sometimes many such events of these chromosomes combining in all sorts of strange ways, and the copy number gains here you can see are very, very significant. There's many of them, right, on chromosome 8. In addition to many more just simple polymorphisms, point mutations, and small insertions and deletions. And that probably has something to do with the fact that many cancers have the DNA repair and replication machinery in many cancerous cells has been hobbled. It's not as efficient, or as it doesn't reproduce the DNA, copy the DNA with such fidelity anymore. And so, of course, our genomes are very big at 3.6 billion base pairs. So even a very low error rate can result in tens or hundreds of mutations after a few cell cycles. And that's a good point too, is that in cancer, these cells are often highly proliferative, so the number of cell cycles, cell divisions, is elevated, which increases the amount of copy numbers, variants, and single nucleotide polymorphisms. And this is an area that I'm very interested in as somebody who's worked on the genomics of cancer and the informatics of cancer. In essence, it seems that cancer uses genomic instability. We could call that all of this together as genome instability or genomic neoplasticity. It's neoplastic because it's changing constantly, as a sort of machine learning algorithm to learn how to do new things. And there's a great paper, classic paper really, called The Hallmarks of Cancer that I put in the required reading, it's in the suggested readings. And there's there's a follow-up, there's 1 and 2. And if you want a quick introduction to cancer, I would highly recommend checking out these 2 papers. The other thing, if you're interested in cancer, is to read The Emperor of All Diseases. And it's a very accessible book that the public could read it too, non-experts in biology. Here I've shown, this is a small diagram showing how breast tumors progress. I'm mostly interested in invasive ductal carcinomas and in situ ductal carcinomas. So this is a duct here. Of course, the main physiological role of the breast is to produce and secrete milk through the nipple. What you're looking at, this is a duct, and these are epithelial cells with some other stuff in them, stem progenitor cells, etc. And the milk travels down this duct here. What happens is that, or what's believed to happen, is that the epithelial or other related cells begin to, there's tumorigenesis. In other words, the genomes become unstable, and those cells start to look hyperplastic, so that means that they're growing faster than what you would expect, and they start to take on morphologies underneath the microscope that do not look like normal epithelial cells per se, and they grow into that duct, and that's what you get is an in situ cancer, where you see this, and it gets filled with cells, and at this point it's not really considered a life-threatening disease, but it is cancer. It hasn't escaped the duct yet, but it is cancer in the sense that these cells are now morphing, and that all comes down to the events that we just talked about. These cells have elevated risks of both SNPs and CNVs, and that process, like I was describing previously, is a bit like a machine learning algorithm, and they're learning how to do all sorts of tricks, like evade the immune system, and to recruit nutrient, and to get rid of its garbage, all sorts of things. And eventually they learn the trick of how to get out, or to invade into the surrounding tissue. So they break through the basement membrane of the duct, and now they're an invasive cancer, and it's now life-threatening. Eventually, then the next step is that it not only gets out of the breast out of the duct into the breast, it now escapes the breast and starts to metastasize by extravasculating into our bloodstream and lymph system until it can colonize new sites, and then it's very dangerous. All of those tricks that it learns that are described in the Hallmarks of Cancer paper are learned basically by modifying, well, I believe, I mean, it's not proven, I should say, it's a hypothesis that those tricks are learned by modifying the underlying genome. It's that neoplasticity that acts as a sort of machine learning algorithm. One of the beautiful papers from 2012 in Cell was from Nick Zano group in Cambridge, and the life history of 21 breast cancers. So they looked at 21 breast cancers, and they applied DNA-seq. So they sequenced the whole genome of these tumors to great depth. At least at the time, it was considered very great depth. It was a very expensive study. And what you see here is, for example, here's a trisomony on 1q. So it's a region of, this is chromosome 1 here. So a region, the Q arm of chromosome 1q, has been amplified, okay? And you see, for example, here, this region on chromosome 4q, that's the right arm of 4, has been lost, right? It's down to monosomy. And there are a lot of smaller events in here that you can't see in terms of polymorphisms, but you can see there's more loss down here, and more loss down here. So a lot of things have gone on in this one tumor. Different regions of the genome have been lost, and presumably there's a loss of heterozygosity in places. And here you can see this is the number of reads that cover each base. And of course, with DNA-seq, like our metagenomics, when we talked about metagenomics and DNA sequencing there, the number of reads is basically going to be proportional to the whether it's the monosomy or trisomy, et cetera, if there's been lost. So here this is monosomic because the number of reads in this region has dropped, and some of these guys up here, now the number of reads has increased significantly, so that would be a sign that there's been amplification or gain in those regions. Okay, so now that's interesting, but what's maybe more interesting is what you can do with this information. Okay, so this is just a restatement that there's been instability, that the genome has evolved, but it's these two figures I want to focus on. So what Nick Zano created were phylogenetic algorithms, the topic of today, to order these events, right? And to find those events, like this trisomy in 1q, that are the earliest events. And then to annotate on these edges from the root down the different events that occurred. So here they're ordering the events by time. Now, of course, we only see what they have is just the cells that exist in the tumor at some time point, right? When they sample by biopsy or surgery. They're using all that information to try and guess the order, right? The phylogeny of what occurred first and what followed. And you get to a node like here, and it branches into distinct subtrees. And you could ask, well, what are they? And that's the concept of subclones in a population. So at some point here, what happened is that some cells went one way, other cells went this way, and the other cells went another way altogether. And that's because in some cells of the tumor, certain mutations occurred, and they were quite successful, and it leads to this new path down here. And other mutations occurred that were successful too, but in a different place from that progenitor population, and they go down, and then this keeps going, right? So this is maybe a more cartoonish version of it, but the idea is that molecular time, that's the tumorigenic event, that's when it started. There's a few cells, one cell there that has some kind of event, whether it's environmental or mistake, or just by chance, or it's genetically prone, for example, to becoming neoplastic. It occurs and starts to expand, and then other events, like this star here, is used to represent other events, like, for example, these kinds of point mutations along here, PIK3CA, TP53, etc. And then those cells become very successful, and they expand, and then other cells come in, and now, for example, here you have green showing up inside the yellow. That means that two subcolonies are coexisting. So in that yellow population, there might be 1 million cells there. One of them has a certain advantageous mutation that seeds this green colony, and that green colony in turn seeds a dark green colony, and some of these cells, like this guy over here, may die out. This orange dies out eventually. Other ones expand and take over because they're just more successful, and you keep getting these de novo genetic events, right? These SNPs and CNVs that keep arising and expanding. Now, just like sequence evolution, there's the evolution part of new mutations, but then there's selection. And of course, in cancer, some of these selective events, like, for example, maybe we could ask the question, why does this orange colony die out, but the green one, the dark green one, expands? Well, perhaps at this point here, the patient, for example, was given chemotherapy, and the orange cells were not capable of surviving in the presence of the chemotherapy, but the green ones were, right? So it may be that this colony here, that tiny little brown colony, was created, and at first it started to do well, but maybe the immune system wiped it out. So there's what are called selective sweeps, and that's the term that we would use, which is basically selection, right, in our evolutionary terms. And so what we're really interested in our lab is understanding and trying to infer this tree. I mean, we can't follow tumors at this point all the way forward. All we can see is the time that we sample cells from these guys down here at time of diagnosis. We take a biopsy or surgery, and that gives us some cells. The computational challenge that we're talking about today, using the type of models we talked about last class, is to then build this, what happened in the past. We're trying to do a sort of molecular archaeology, right, to look back at what happened. Now, if we can understand and estimate this guy back here, maybe we have a dramatically different way of treating cancer, because perhaps it's the trisomy 1q, for example, or these mutations here are what really make what start cancer. Maybe it's those things that we should target prophylactically, right? So And intellectually, it's just super interesting to know exactly how do cancers arise, because we'll never really be able to. Well, I shouldn't say never. We can't show that, but right now it's still extremely challenging to imagine how we would ever be able to, for example, find that one cell or handful of cells in a woman's breast that has decided to become cancerous, right? Become neoplastic. It's not until it grows to a relatively sizable proportion, maybe a billion cells, right, a cubic millimeter or something, that something like mammography could pick it up. By the time you can feel a lump in your breast, that's an awful lot of cells, right? But we would like to have some kind of indicator very early on. Ideally, as soon as a cell starts to commit to instability, we would like to target it. We're a long way from that. Makes this one of my favorite topics in science. So physicists use telescopes to look back in time, and phylogenists use computation to look back into the beginning of evolution of life on Earth. It's all about this tree here, rooted at the ancestor of bacteria, the archaea, and the eukaryota. This is all cellular life on the planet that we know of to date. It could be more, but it's drilling down into these trees and subtrees and sub-subtrees, etc., down into the different fungi and animals, plants, and all sorts of different bacteria, single-celled organisms, and understanding how we evolved, right? How life expanded on Earth, and all the different forms that it takes. Okay, so when we have such a tree, now usually it's not the entire tree of life, which actually you guys have downloaded and played with on assignment 3, so you have a pretty good idea of how big that is. We have different objects. One is the internal node. The internal node has children, or it has more than one. It has more than one adjacent node. So this guy here is internal because it has 3 adjacencies. This has 3 adjacencies, etc. But these guys down here have only one adjacency. Okay, this one, this one, this one, this one, and these 2 here. We call these leaves or external nodes. Okay, and we think of the leaves as being generally extant species, so species that are existing today, and the branches, these, or we call them edges more often, or I call them edges more often, that represents evolutionary time, the time that it took to evolve from this node to that node, or from this node to that node. In this case here, this tree is unrooted. There's no, we don't, unlike the tree up here of life, we know that bacteria and archaea split, and then archaea and eukaryota split, so we can root that tree. This tree is unrooted, so we don't know the direction of evolution. Perhaps the root existed along here, or perhaps the root existed up here on an internal edge. So here would be the same tree, but we root it, say, at this node here, and it suggests that the yellow evolved after the root of this tree, and this direction became yellow, and this direction stayed brown. We can think of the root as being the least common ancestor of all of these leaves here. Okay. We think of the internal nodes as being ancestral or extinct, that we don't actually see directly. They're inferred, right? So this is, we're guessing at what those internal nodes look like, much the same way that we look at bones of a dinosaur and guess at what it looks like. We're looking at present day DNA or amino acid sequences and try to guess what the ancestral sequences look like. Or if we're not using molecular sequence data, we might be looking at here properties like color, beak length. You can see that these all differ a little bit this guy has a red hat, this guy has a strange throat thing, this has a long tail, etc. And these different features or attributes of these organisms have evolved as this tree progresses. The other concept here is the idea of an outgroup. So if we root the tree, we would like to know, if we're looking at this, if our study is mostly about these yellow birds, then we would like to know the direction of evolution. So what we often do is pick a species, in this case here the brown birds, that are clearly quite far away evolutionarily. And what that does, these two extant brown birds act as then the outgroup, and we know then that the evolution across all the yellow birds goes in this direction down from those from that root here. There's different kinds of trees that we can think about. The simplest might be the cladogram, and the cladogram is not weighted. So these edges here, they could be rooted, so we know the direction of evolution, that yellow and we don't know if it was yellow or brown at the top here, but we know that it's a deciding attribute between those two sub-clusters, or sub-trees, to be more precise. But we don't know anything kind of quantitative about this. It doesn't tell us anything about the timing. These edge lengths don't mean anything. And we don't really know how much those species have diverged. It's sometimes convenient to put on here numbers that represent, for example, time or the extent of divergence. So, for example, this might be a measure of PAM. So this would be saying that for every 100 amino acids, we would expect 3 point mutations. And this edge here, it's longer because from this period to this period, we expect 6 point accepted mutations per 100 amino acids. So those, generally speaking, it's much more informative to know not only the genealogy of how these different observed taxa or species branch from each other, but also some measure of how much evolution there's been or time. Okay, so we've talked a little bit before why time is difficult on these trees, but an ultrametric tree makes it simple. So an ultrametric tree, all the leaves are the same distance from the root. Okay, so that means that you can think of this as a kind of constant mutation rate along all branches. So the distance from here down to this species here is the same as the distance from here to here. And so basically the rate of mutation has stayed equal over the evolutionary period that we're looking at. So we can think of it as a molecular clock that ticks regularly. And we know that that's problematic because of things like selection that would make it very unlikely that the rate of accepted mutations, which is what we observe, right? We don't see unsuccessful, non-accepted mutations. We only see the accepted ones. It's hard to see how that could correspond to time. But nevertheless, the ultrametric tree is an important conceptual object. A bit less restrictive and more common is the additive tree. And I don't really think that we need to have a definition of that. It's the distance from the root to the leaf can vary across your species, which actually makes sense because, of course, there may be different rates of evolution in the yellow birds versus the orange bird because they live in different environments. It can be all sorts of things where that for some reason there's more accepted mutations over here because the environment is more forgiving than over here, and that's actually that's a well-documented properties of many evolutionary contexts where the pressure, the selection pressure being placed on populations dictate to some extent how many mutations are accepted. We won't go into that too much. So in all of these examples here, there's a sense of evolution by descent, meaning that there's time or PAM, but the evolution is sort of flowing down this tree that mutations are going from parent to child, right, over time. But that's certainly not always the case, and it's certainly not the case, maybe even often the case in bacteria and archaea and other single-celled organisms, perhaps also fungi, for example, single-celled fungi, or algae. I'm not quite sure about algae, but it would make sense. So, in fact, the definition of species in bacteria and archaea can be quite challenging because they pass so much information laterally, lateral gene transfer. Sorry about that. So basically, two organisms living in close proximity, in fact, maybe even being engulfed by the other organism, or in some kind of endosymbiotic relationship or ectosymbiotic relationship, they might pass genetic material, including genes, and that's been well documented. There's many cases in the literature where they can pin down to genes that have been transferred from parasite to host, and that includes from bacteria to the eukaryota, right, to animals, plants, and fungi, but often between different species of bacteria. And we won't get into that today. It's an area where I used to work. When I first started out as a postdoc, I was working quite a lot trying to design algorithms to detect lateral gene transfer events. And on the other hand, away from bacteria, but especially in higher order eukaryota like human, you have a lot of gene duplication and loss. Well, it's actually incredibly true in plants, and they have a lot of duplication and loss. And this leads into the concepts of orthology and prology, but one thing at a time here. So we say that two sequences are homologous if they share a common ancestor, which is kind of vacuous, because in a way then, if you believe that we aren't coming from Mars, or we haven't been seeded more, life hasn't been seeded more than once, then in fact all sequences trace back to one common ancestor. It gets used as slang a little bit too much in the literature when people actually mean high sequence similarity or statistically significant sequence similarity. But it really just means that two sequences share the same common ancestor. Now, more precisely, two sequences are orthologous if their common ancestor is a speciation event. Okay, so basically, when, for example, let's take here a gene. Let's suppose this is a gene tree for some specific gene, maybe a ribosomal subunit, for example. Then the gene in chicken and the gene in human, its least common ancestor is here, and that means that in the ancestor of human and chicken, we can trace it back to the ancestor of human and chicken. Another kind of event is the gene duplication, though, and this really confuses phylogenies. And we won't go into this more today, but this is just for your interest. So sometimes genes get duplicated. So here in this figure, you can imagine this as being this black tree out here that pipes. Think of it as a species tree. And in the species tree are all the different genes in the organism. This is one such gene, this blue line. And that's one gene tree. So this might be one ribosomal subunit that we're going to follow over evolutionary time. And I should say in this direction here, it's time. Okay, so as you go down this tube structure, you're going down time. Okay, and there would be multiple lineages here, multiple gene trees that would go down this trunk, one for each gene in the genome of the organism at this point. That's marked by a blue dot there. And so as this organism evolves, at some certain point a gene, or genes, a whole chromosome even, but here just a gene gets duplicated, and now there's two copies of that gene at the same time in that genome. And so it's the least common ancestor of this gene, and this gene is rooted back here. And we would say that they're paralogs. So they arose from a gene duplication event. And we talked yesterday about gene duplications as one of the types of genomic aberrations that occur, or even common. It's a common occurrence in most of our genomes. So basically, now these two genes evolve independently within that organism. And if you think about it, it's the fact that you now have two copies of that gene, or for example, since we're diploid, we'll now have three copies of that gene. It now gives us sort of evolutionary wiggle room. Now the organism has a bit of space to play, right? Because whatever that gene did before, well, the two copies obviously suffice because the organism was alive, right? It functioned. But now that it has three copies, well, it's not so reliant on any one of those copies. And so gene duplication is believed to be one of the major drivers of novel gain of functions in organisms. The ability to copy a gene and then let it evolve because it's not under tight selective constraints really opens up a way for organisms to discover new functionality. But in this case here, this duplication, that bar indicates a duplication event, and they co-evolve until we get a speciation here into something that leads to the pipe that leads towards species A, and a pipe that leads towards the ancestor of two species, B and C. And what you see here is that this gene now has a copy in this lineage, and it has a copy in this lineage. Likewise, the second copy of the gene has a lineage this way and a lineage that way. Now, the second type of event that we observe is gene loss. So maybe because that gene isn't needed, the second copy in this lineage here, there's no reason for it, and it just evolves away from functionality, and we say it's lost. But this gene, we can't get rid of it, so it stays. And now what happens is that organism down here called A has one copy of the gene. Now, on the other hand, this lineage goes all the way down, and at the ancestor of B and C, both copies of the gene are present, one from here and the one donated from there. But after that, when we get down to organism B and C, this one is lost here, and this one is lost there. So you might stop the video right now and you think, okay, well, ask yourself, which pair, AB, AC, or BC, are closest, those genes? Which ones have the least amount of evolution between them? Well, the answer would be that A and C will have the least amount of evolution between them. Why? Because if I trace A to C, I go back up here to this event here, and I trace it down, and basically I hit C here. Not basically, I do. So the total amount of evolution is this plus that. Now, if I go from A to B, I have to go all the way back here like before, but now I have to go all the way back up here to this duplication to come back and go down to B. Likewise, for C to B, I have to go all the way back up to this event here, back up through the duplication, and down here. So although species B and C are the closest, because the other genes in the organism, the thousands of other genes, don't have such a duplication, when I look at just this one gene family, I get the wrong idea of the phylogeny. The phylogeny I have would look like A and C are close together for that gene, but actually overall it's B and C. We're not going to talk more about these questions of gene duplication and loss, but there's significant conceptual and computational challenges involved in dealing with those very interesting genomic events. What I wanted to talk about today are two different methods for inferring phylogeny. The first one is really simple to understand, and it's not at all based on Markov models or any kind of probabilistic model of evolution. It's called parsimony, and it's based on an important concept in science called Occam's razor. Occam's razor shows up everywhere, I would say it shows up also in our day-to-day life, right? In our interactions with other people, all sorts of things. It basically says the best hypothesis is the simplest hypothesis. It's like the KISS principle of the military, right? Keep it simple, stupid. So we're not going to postulate anything to explain our data that's superfluous. We're just going to, boil it down to the bare bones. So for phylogeny, this translates to building the best phylogenetic tree is the one that requires the fewest changes. Okay, so what is the phylogeny problem? Well, our objective is that for a set of input sequences that would look like this, okay? So we have 4 species here, alpha, beta, gamma, delta. Just to make sure that you know, that's gamma. And I think the other alpha and beta are okay. And this is delta, okay? 4 species. And let's say this the protein that we're looking at is really short, just 5 sites. So that's our input. We have an alignment of those proteins. We would assume that these 4 proteins are coming from orthologs from the the 4 different species. Our goal is to find the tree that requires the minimum number of substitutions to explain the observed or inferred differences between sequences. Okay, that tree, which requires the minimum number of substitutions, is the most parsimonious. Okay, so that's a bit wordy. So let's look at this here. My hypothesis here, or my statement here is that there are 4 possible unrooted trees. We have alpha and beta together here, and gamma and delta on this side. That's one possibility. The second possibility is alpha and delta on this side, leaving gamma and beta over there. And the third is alpha and delta. So alpha beta, alpha gamma, alpha delta. You can ask yourself, you can stop the video and ask, are there any more trees, possible trees, on 4 leaves, unrooted trees, okay? And you should convince yourself there's not. Okay, so now we have these 3 trees, and we have our input data, and the question is then, now, is which of these 3 trees is most parsimonious? Okay, so let's see here. Let's take a look. So the first thing here is that if we look at site 1, okay, this is site 1, it's all L's. So that's really easy. This is an easy case. This is completely uninformative. Why is it uninformative? Well, if I put L on these 4 leaves, right, in any of these 3 trees, what is my score? Well, if I have an L down here and an L here, and I put L there, there's no mutations observed, right? 0 mutations there. L to L is 0. I have L here, and I have L there. So now I can put, for example, an L here at the internal node. There's no mutations there. There's no mutations there. And there's going to be no mutations between those 2 guys. And so my score for this tree is a 0. Okay? There was no penalties at all. And the same thing is true here, and the same thing is true there. My scores are always equal to 0 for all 3 trees. And so this site, because they're all the same nucleic acid, amino acid, sorry, it told us, it doesn't give us any hints about what the phylogeny would be for these 4 species, right? That hopefully makes sense. So I'm going to quickly erase those guys, and let's look at the second character. Okay, so the second character tells me that I should put W on alpha, P on beta, I on gamma, and R on delta, right? Now, so now it's a bit tricky, right? Because all 4 disagree. So it probably wouldn't make sense to put a leucine there, because then I would have a cost of 1 on this branch and this branch, right? That doesn't seem right. So I would probably choose, for example, either a W or a P. Let's say I choose a W, okay, the tryptophan. That means I score 0 on this branch, no mutation. I score 1 on this branch to switch from tryptophan to proline, right? think that makes sense. Now over here, the same logic, but let's put R there. And it won't matter if I use I or R right there. It doesn't matter. That means there's a cost of 1 on this branch, no cost on the R to R. Finally, I get a cost here to switch from tryptophan to arginine, and so now my overall cost is 3 on that tree. In fact, I'll leave it to you, because all 4 amino acids are different, if I put them onto these trees, or this tree, I'm always going to score 3. You can verify that in your own time. So actually, that, because they're all scored 3, that character is also completely uninformative. It doesn't give us any hint about the phylogeny of these 4 species, because the character just keeps changing, right? So that's not very interesting. Well, what about site 3? Okay, so it tells me to put an S, a serine, on alpha and beta, and an A, I guess it's asparagine, on gamma and delta. So that would be an S here and an A here. I think I probably mislabeled in the previous example. I wasn't following, or I wasn't being very careful, but the W, P, I, and R from the previous site has to match up with the labels on the leaves. But it doesn't change the story that you always have a score of 3. I'm sorry, that was a bit sloppy of me. Delta is an A, and beta is a S. Okay, in that tree. Now you can already probably start to think about that as I finish this guy. That alpha is an S, beta is an S, gamma is an A, and delta is an A. So now this is interesting, because if I go back to this guy here, if I have an S at alpha and an S at beta, then it makes sense to put an S here. And I have an A and an A there, so I put an A there. So the score here is 0, 0, 0, and 0. Good so far, but I'm going to incur a score of 1 to switch between S and A. What about over here? S and A, well, I don't know what to put. I could put an S there. It's arbitrary. Again, over here, S and an A, I could put an S. That would make sense, because this guy has an S. And so the score is 0, 0, 0, 1, and 1. So my score overall is 2, right? So which tree is better, this tree or this tree? And the answer is clearly that this tree right now is better, right, by a score of 1. And what about this guy down here? I think it's the same thing, right? I could put an A here and an A there, and I'm going to get a score of 2. But I can't figure out any way to make that lower than 2. And so on this node, it's going to, this node is informative. That's the first thing. And it favors the first tree, right? It favors that tree. Okay, so that one favors that tree. The first 2 didn't favor anybody, so let's go and do the last 2. Just clean it up a little bit, guys. Bear with me. Whoops. Yeah. Okay, I edited things out for you, so you didn't need to listen. And now I'm labeling by the 4th character. So follow with me here. That'll be an R, and I'll have an S down here. So right off the top, when I have an R and an S, R and an S, I know this is going to cost me 2, right? It's going to cost me 1 and 1, so that's going to be 2. Alpha is an R, beta is a S, delta or gamma is a R, and delta is an S. So I'm going to have a score of 1, right, for this guy. And so so site 4 is looking pretty good. Down here, this guy is R, S, R, S, and we know that we're not going to do any better than 2, right? So this site here favors this tree. So site 3 favors this guy, but site 4 favors this guy. Now you can see the difference between 4 and 5 is superficial. I replace R by Q, and S by T, and we know right off the top that it's also going to favor this tree. So the most parsimonious tree for this matrix is this tree right here that puts alpha and delta, alpha and gamma together. Okay, I've left a bit of space here on the next slide so that you could work through this yourself. So what you should do is just, turn off the video and see if you can reproduce what I have just done. Okay. All right, so the problem is that, well, for 4 leaves, man, that's no problem. Like, I just ran through all 3 possible trees, right? That's okay. But what if I had 5 or 6 or a billion leaves? Would this be feasible? So this is not, I just want to show you, I'm not going to test you on this, so there's no reason to get stressed out. But I'm going to show you it's completely infeasible. So imagine I asked the question, if I wanted to go from 4 to 5 leaves, how many different trees would I have then? Well, if I have four leaves, here's one of the 3 possible topologies, right? Okay, we understand that's one of the 3 possible topologies from the previous example. And I could put the new edge here, or I could put the new edge here, or I could subdivide that edge, or that edge, or that edge. So basically, no matter where I put the edge, right? Wherever I put the edge, I create this edge, I subdivide this edge, and I make 2. Okay, so I a new edge there, a new edge there, but I lose that complete edge, and I get this edge here. So overall, every time I subdivide, I get 2 new edges in total. Right? Okay, so you should convince yourself. Stop the video and convince yourself you understand. So now, if I have 2 leaves, I have 1 edge, right? That just looks like, I mean, that's easy. Just if I only have 2 leaves, then I only have 1 edge. If I have 3 leaves, then I have something like this, right? This, right? I have 3 edges. If I have 4 leaves, I had this scenario up here. That's 5 edges. And 5 leaves is 7 edges, etc. So you just keep adding 2. That's the first thing to understand, is that if I have n leaves, I'm going to have n plus 2 edges. Okay, so now, and the new node can be added to each edge of the tree. So those are the 3 properties. When I add a new edge, I add a new node, I add 2 new edges. That's property 1. In total, I have n plus 2 edges if I have n leaves, and there are as many ways to add the new node as there are edges in the old tree. So for an example, here the old tree is 4 leaves, and I'm going to 5 leaves. So now, what does that mean? So a bit of arithmetic, nothing tricky, but when I want to go from 4 to 5 species, I had 3 trees, so I could do this to each of the 3 trees, but now there's 5 ways to add the edge, okay? So I have in total 15 possible trees on 5 leaves. Now if I go from 5 to 6 species, I have 15 trees, and they have 7 edges, right? N plus 2. 5 plus 2 is 7. So each of those 15 trees can be modified in 7 ways. Okay, and so total I get 105 trees. From 6 to 7, I get this 105 times 9, so 7 plus 2, that's just under 1,000. Then from 7 to 8, I take that 945. Now I go from n plus 2 is 11, I'm up to 10,000. And by the time I get to just 15 species, I have, what is that? That is 7,900,000,000,000,000 trees to consider. And that is no longer feasible. Now, you know from assignment 3 that the NCBI's taxonomy had something like several thousand nodes. So if you were to try to build a parsimony tree on several thousand nodes, that there would be more possibilities than there are molecules in the universe. So it's completely impossible to solve parsimony by simply enumerating every tree and checking how much it costs, like we did in the example. I give the example because it's interesting and it's important for you to understand the type of argument we're using. the computational scientists that come in, that are coming from computer science, math backgrounds, build heuristics that try to find good trees efficiently. And that's really what computational biology in computer science is all about. How do you search these giant spaces in an efficient manner? I mean, I think you probably have some insight into that now. The other problem with parsimony, it's limited because it doesn't have a realistic model of evolution. Why does that matter? Well, here's a little thought experiment, okay? Imagine that this is in fact the correct tree. Let me explain this tree. It's pretty interesting. And this is something called the long branch attraction problem. So the real tree, the 4 species are like this. They have long branches. These branches now, think of them as PAM units, okay? So there's been a lot of evolution, a lot of iterations of the Markov model between the ancestor of delta and beta, okay? That guy there. And there's been an awful lot of evolution from the ancestor of delta and alpha on the way to alpha, right? So this branch is very long. So you can think of it, maybe it's been very fast evolution, like after the Cambrian explosion type thing. But from here, from delta to its parent, there hasn't been much evolution. Maybe it's been, these organisms have been living in very constrained environments, and so it hasn't been able to sort of, let's say, poetically, spread its wings and evolve. And the same thing for delta. It hasn't had much evolution. So the problem is that parsimony will not favor this tree, right? It would favor this tree down here, because it would put a P here and a Q there, and the score would be 1. So so to recap, we know, right, that the rate of evolution does change over time. When you get this kind of effect where these long branches exist after speciation, parsimony wants to put them together, but they don't belong together. Okay, so I think maybe you could stop the video and think about that. If you don't get it right away, don't feel bad, because I think a lot of molecular phylogenists struggled with that concept for about 30 years before we've sort of figured it out and understood why these methods fail. But we can go back to maximum likelihood, which is something now you guys are really becoming experts on with Markov models. So basically, in the maximum likelihood model, it goes back to that old log likelihood now that we've seen in several contexts, from binding sites and gene finding, where we want to evaluate the data we've seen, the probability of the data we've seen, given a model of evolution, versus the probability of our data just sort of spontaneously evolving randomly, right? So background, that's our background, right? This guy down here is our background distribution, and this guy here is our model of evolution. And so when there's more evidence for the model, this log ratio is going to be above 0. When they're equal strength, they'll be 0. And when the random model is stronger than the model, it'll be negative. Okay, so let's do this. This is going to be a bit tricky, I'll warn you, but several concepts going on here, but I'll try to break it down step by step to give you the intuition. Okay, so let's suppose our model is described by a tree with PAM distances on the edges. So here we have alpha and beta together as siblings, and they're separated by 105 PAM units. Recall that PAM means we expect 105 point accepted mutations every 100 amino acids, right? And the ancestor of alpha and beta here to the root is separated by 40 PAM units. So think of it again as 40 iterations of the Markov process. Likewise, we have 30 to 45 and 60 for these two guys. So we call it a model because we can kind of imagine starting here at the root and simulating with our Markov process the evolution that leads to alpha and beta through this through this speciation event. Same thing for gamma and delta, of course. Okay, so now we see these sequences here at the leaves of this tree, right? So this is our input. We have actually the alignment of our protein that we're looking at, amino acid sequence, and I don't know, God gives us a tree, a weighted tree that describes the evolution. This is a putative tree. This is a hypothesis. It's a one model, but that's where we start with maximum likelihood. We sort of turn to God and say, I don't know anything about these sequences. Give me a model, right? Give me an idea. Okay, so what do we do with that tree now? Okay, so the maximum likelihood approach maybe is a bit counterintuitive at first, but makes a lot of sense actually in the end. So there are really three unknowns in this game. There's this X. So which amino acid, okay, I should say there's three unknowns for each site. So let's look at site 1. L, 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 and L. Okay, that's at the leaves, right? And we can repeat this for all of them. But for this first site, there's three unknowns. Which amino acid existed at the root that led to Y and led to Z on this side? Okay, as an interesting thing here, ask yourself, what would parsimony do here? Well, parsimony would put an L and an L and an L and say, I score 0 and I'm done. But we know, because we're Markov model experts, is that there's no way to be certain that there was something else like an isoleucine up here at the start of time that mutated, maybe became a leucine, and then maybe it became an asparagine, and then it became an alanine, but eventually it mutated to an L. And especially over these long evolutionary distances like 80 and 60, it's quite possible that substitutions between leucine and isoleucine occurred silently. We don't see them, right? They come up, but as long as it ends up at after 120, units of evolution as a leucine, we don't care. Maybe it was just dumb luck. Maybe it's just the total luck that we saw leucine at 4 nodes. That could happen. There's 20 amino acids and they all have frequencies. They could just show up as leucines, and that is exactly the spirit of our background model. We would just look at the frequencies of our four leucines showing up in a row, essentially, right, over the 4 species. Okay, but now in total, for the first position though, in the maximum likelihood, we consider all possible scenarios. So there's 20 different amino acids that could go here, times the 20 that could go there, times the 20 that could go there. And so our approach here basically sums up the probabilities or the likelihoods over all of these 8,000 possible, 20 times 20 times 20, I believe is 8,000, right? 8,000 different scenarios for the first position, right? Already, I think you can start to see how computationally cumbersome this problem will be, because I have to evaluate something for 800 times just for this first site, and then do it again for every other site, not to mention some stuff to come. Okay. All right, so let me give a bit more concrete idea then of what I mean by the model here. So what I'm going to do, right, is basically sum for all possibilities of X. That's going to be 20 different amino acids. I'm going to say basically hypothesize and say, I believe an asparagine is here, okay? And then I'm going to hypothesize it's an isoleucine, and then a leucine, and then a tryptophan, and then blah, blah, blah. And for every possible choice for X, I'll consider every possible choice of Y. And for every possible choice for Y, I'll consider every possible choice for Z. So that's why you get 20 times 20 times 20, which is 8,000. Now for each choice, I don't care what it is. It could be I don't know, let's just pick something at random. Proline, isoleucine, and leucine. That's one of the 8,000 choices. Here's what we compute. We come down and we say, for my choices of proline, isoleucine, and leucine, what's the prior probability of X of proline? It's quite rare, so that's going to be a small probability. And then I'll compute the probability that X switches to Y. In other words, proline switches to isoleucine at PAM 40 over 40 units of evolution. So where would we get that score? Okay, where would we get that score from? And the answer is that we know that exactly because we would use the PAM 40 matrix that we have. And that would tell us, we would look at the Y column, X row, because remember they're reversed in the PAM matrix. And that would tell us the probability, or the log odds, log probability, of Y switching to X over 40 PAM units. And that's this branch. And then we would also multiply that by this event, that proline switches to leucine, but this time we would use the PAM 30 matrix, a shorter evolutionary time. That's this, times this event, Y to L. This is in alpha. I should have marked that probably. This is Y to L in alpha, but the 25 gives it away. And this is Y to L in beta, right? And that's 80 PAM units here, right? Okay. Finally, we look at here, we calculate now the probability of going from Z to L across 45 PAM and across 60 PAM, Z to L. So leucine to leucine, leucine to leucine. Presumably these would be quite high scoring, right? Because the leucine has stayed the same. Now over those 8,000, that's our likelihood for this one tree. Now this is one tree. Now remember, we asked God and said, okay, well, God, what is the right tree to use, right? And God passed us this one, but it's not the only tree. In fact, well, we know that there's basically 3 possible trees, right? Well, the root's kind of problemsome because I can root it in different ways, but let's forget about that for a minute. But there's different ways to different trees, but there's infinite number of ways I can weight that tree. What if I change 40 to 41? I'd have to recompute everything. What if I change 80 to 800 or 700 or 6? There's an infinite number, uncountably infinite number ways of just changing the weights on this one tree, never mind considering all different trees. Okay, and that's the source of massive computational challenge, right? How do you find over all possible trees the best tree, the one that maximizes that likelihood, that equation we just looked at? So this is one tree here that we just looked at, but I could look at the other trees, the 3 trees that we had for parsimony, alpha with beta, alpha with gamma, alpha with delta, but even then I would have an infinite number of ways to reweight those trees, right? So how do you find the maximum likelihood tree? You would have to look over all of these possible scenarios, and there still to this day is no real efficient way of finding that. It's provably a hard problem in computational terms, but there's a lot of good software out there with good heuristics that does a pretty good job of finding good maximum likelihood trees. And as a side note here, the maximum likelihood approach for phylogeny, with perhaps its cousin, the Bayesian approach, are the best ways of doing molecular phylogenies. Let's recap here. We have models of evolution, whether at the nucleic acid level or the amino acid level. If it's coding, we want to work at the amino acid level. And those PAM matrices, or BLOSSOM if you'd prefer, they're incredibly powerful ways of estimating the probability that some amino acid flips to another amino acid over an evolutionary distance, right? Those matrices harbor all of this, these different paths through the Markov chain to go from A to B, right? Over a certain number of iterations in the chain. And I think by this point in the course, you're sick of hearing about Markov models, but they're so powerful that you need to get them kind of just second nature inside of you. So the other way you can, one other way, not the only other way, but another way that you can use these Markov models is in sequence alignment as a way of scoring. Okay, so what is sequence alignment? Okay, well, first to say here is that this problem has been looked at just massively by the community for a long time, and BLAST is is sort of the industrial strength approach, and the NCBI supports BLAST, and can't go through your career without using BLAST. Now, it has many more bells and whistles, and it's been sped up because these databases are huge, and we're not going to go into all the BLAST theory. It's very interesting, actually, how it works. But it's based effectively on what I'm going to show you today. Aki will try to lead you through in the lecture of how to use BLAST in practice, right? It's quite simple. I mean, if you understand this, going and using it at the NCBI is like trivial. But I wanted to show you a computational technique called dynamic programming, because once you understand that, then a lot of doors open up for you about how a lot of bioinformatics software works underneath the hood. And that's important, again, because I think when you're using all these different packages out there, whether it's in alignments or Markov models, etc., you have a better sense of how to set these things up properly and use them properly. You're not guessing as much. This is an example of an alignment, just a random one I downloaded from the web here. I think it's aurora kinase A versus I'm not actually sure what that is, but there's two different proteins. This is a fragment of the protein. And you can see here that each amino acid are aligned vertically. And it's because that the person who wrote this paper believed that this amino acid in this protein evolved into this amino acid in this protein, right? So they're in this, let's write it this way, is that I have my species A and my species B, A and B, and presumably I'm looking at the orthologs of these two proteins, and I assume essentially that there's some ancestor to species A and B, and there's been T1 units, like PAM units of evolution leading to A, and simultaneously there's been T2 units of evolution leading to species B. And in the first position we have an L and an A, and the question then is what amino acid existed up here at the root, right? And the same thing repeated for every other amino acid in the chain. And so the alignment is supposed to represent amino acids that evolved from each other, right? And so here you have a mismatch, L to A, and there's another mismatch here, V to N. Match, K to K, D to D, all these guys are matched. And then you have these things that are called insertions or deletions. Now, when we have two guys like this, we don't know the direction. Were these guys here, S, D, G, and E, lost in species A? Or were they gained? Was it an insertion in species B? We don't know, right? All we know is that somewhere along the evolution from here to A and here to there, if it was on this side, let's say here, those amino acids would have been lost on the way to A, right? I can write that out here. So somewhere in here, these four amino acids were kicked out of A, and that's why we have a gap. Or, for example, these four amino acids weren't at the root, and they were inserted here, right? So there was basically an insertion of those four amino acids, 1, 2, 3, 4, along this branch here at some point, and that's why they show up down here. Let's rephrase this a little bit, because we don't really, in the alignment, we don't really care about this root. What we want to do is find the best way to align this sequence at A with this one with B, and so we can basically re-express this as A evolving to the root, and then root evolving down to B. So instead of going from the root down and down, we're just going to go this way, see it that way. And so the total evolution between A and B is T1 plus T2 PAM units, right? And we'll see in this first position here, for example, that L has switched to an A, right? That's the equivalent to saying that T2 plus T1 PAM units separate B to A, if we want to go in that direction, right? And we went from A to L. Actually, I forgot that in the PAM matrix, see, I do this all the time, that in PAM matrices, it's reversed, right? Oh, how does this work again? I hate this. I hate this problem all the time. I would normally say L to A, so it's A to L, and this would be L to A, right? Doesn't matter. They're symmetric. So it doesn't matter if I go in this direction from A to B or from B to A. It doesn't change anything. Now, does T1 plus T2 differ from T2 plus T1? Well, for you and I, we understand that there's absolutely no difference because, well, addition is like that. 5 plus 3 equals 3 plus 5. Of course, some questionable right-wing leaders in the United States don't understand that. It doesn't really matter what the order the ballots come in. Okay, so that's, of course, equal to, well, not of course, this part here goes back to our likelihood model. It says that I can sum over all possible ancestral states. So I would, like the previous slide, I just ask, what's the probability of going from L, and then I try every amino acid there, and I sum over all possibilities, and of going from A to L, A to A, S to L, S to A, over all possible amino acids, X to L times X to A, and that's my likelihood. Okay, so next class, we're going to pick it up right here, and I'm going to go through the dynamic programming algorithm with a simple example back at the nucleic acid just for convenience, and show you how sequence alignment works. Okay, so I'll see you at next class.